Go.
This lecture is about cell communication. So we're all composed of cells, and the cells have to work together to talk to each other. We know that cells talk to each other in a variety of ways. Even simple unicellular organisms like yeast talk to each other. Amazingly, there's two kinds of yeast. There's A cells and alpha cells. The A cells secrete A factor, which is a pheromone, and the alpha cells secrete alpha factor. When they find each other by these pheromones, they actually schmoo. They actually kind of extend towards one another. And this shape here reminds us of Pogo's character, the Shmoo, from the cartoon Pogo, which many of you probably haven't seen because it hasn't been in print for a while. So why do they secrete pheromones? It's because they have sex. Yeast have sex. They can propagate in the haploid state, or they can fuse with one another and become diploid. And then they do this thing called meiosis and make spores. So they talk to each other using these pheromones which bind to receptors. They communicate with each other. The cells communicate with each other. So what do cells do when they're talking to one another? They can do all sorts of things. Pretty basic stuff. Cell communication tells cells to survive, that is, don't die, or die. You can tell cells to die and they'll die. You can tell cells to divide, that is, go through the cell division cycle, or to differentiate, that is, when cells need to become something else. So cells change in response to signals. They change because the extracellular signal is converted to an intracellular signaling pathway. So what I'm going to talk about is all the stuff that happens inside cells when they're talking to each other through different molecules. Many things happen. Your extracellular signal molecule binds to a receptor that can change a protein's function, can change the cytoplasmic machinery, can change the cytoskeleton, causing cells to move. It can also change gene expression, so transcription factors are activated, or DNA replication is started. So you get different patterns in gene expression. All of this results in altered cell behavior. So this really tells us that cells are alive. Cells are alive. We're all composed of cells. All these living things inside of us working together in various ways. Cells have receptors for specific molecules. That is, they receive information from outside of them, extracellular information, and then activate what's called signal transduction pathways to convert the extracellular message to intracellular information. Signal transduction pathways cause cells to change behavior. That is, change their shape, their very structure, their sense of identity, or to divide or die. In general, signal transduction is defined by a process by which cells respond to the extracellular signal. So the extracellular signal, this little red ball, binds to a receptor. That causes a whole series of molecules to change. Some might be metabolic enzymes to change the metabolism. Some might be gene regulatory proteins to cause changes in gene expression, or cytoskeletal proteins to cause changes in cell shape or movement. All of these involve molecular on-off switches. So a series of proteins are turned on and off by molecular switches. And I'm going to talk about those switches. So what do we mean by molecular switches? Two examples are the main theme of this lecture. One is kinases put the phosphate on proteins. They put phosphates, covalently attached phosphates, PO3, to hydroxyl-containing amino acids on proteins. That is serine, threonine, or tyrosine. The signal comes in. The kinase puts a phosphate on the protein. That's this little P here. That requires ATP, so the third terminal phosphate is transferred covalently to the protein. And phosphatases take this phosphate off. Phosphatase removes phosphates. And that terminates the signal. So it's reversible. Okay? That's an on-off switch. Phosphorylation and dephosphorylation. Another kind is binding GTP. And I'll talk more about that in a little bit. In this case, the protein is not covalently modified. In its resting state, it's bound to GDP. It exchanges its GDP for GTP, and the GTP bound form is active, and it sends the signal out. This is not covalently bound, but this guy is a little enzyme, can hydrolyze the GTP, take off that terminal phosphate, and it goes back to its resting state, bound to GDP. Instead of kinases and phosphatases putting phosphates on and taking them off, we have guanine nucleotide exchange factors, which cause the protein to kick out the GDP, and then GTP can bind. That's called a GEF, exchange factor. And then we have another protein that regulates these molecular switches called a GAP, activates the GTPase, so this phosphate is hydrolyzed. These are two important mechanisms that are used over and over again in signal transduction. So we can characterize or categorize or put in boxes, here's some boxes, for different types of signaling that occurs in multicellular organisms. Endocrine signaling involves secretion of hormones into the bloodstream, which then goes and binds receptors on cells that are far away from the original hormone. An example would be your pituitary gland, which secretes hormones, and they bind throughout your body. Paracrine signaling is like what yeast does. A molecule is secreted by the signaling cell, and it binds to local cells. The molecule just diffuses to the local environment. Neuronal signaling, I talked about in the lecture when we talked about the propagation of the action potential. That's a specialized form of secretion, regulated secretion, that occurs right at the synapse. So small vesicles containing neurotransmitters are released, and then the target cell binds to those neurotransmitters. Some cells signal by direct contact with each other, and in this case, the ligand, or the signaling molecule, is bound to the membrane of one cell, binds to the membrane receptor of the other cell. So when we're thinking about endocrine signaling or neuronal signaling, we have to think about regulated secretion. And that is, molecules are packaged into secretory vesicles, which fuse with the plasma membrane, releasing their contents. The fusion of these with the plasma membrane is done in response to a signal. So some other signal causes the cell to secrete. This is in contrast to constitutive secretion, which all cells do. Only specialized cells have the ability to secrete on demand. in response to a signal. Those would be pituitary cells or nerve cells. So when we think about endocrine signaling, the endocrine cells are close to the bloodstream, so they're vascularized, and they secrete hormones which bind specifically to cells that are the target cells. How are they binding specifically? Because the molecules have to swim all through the bloodstream and diffuse, the specificity relies on the binding specificity between the hormone and its receptor. So that means that the association constant is very high. You'll remember from chemistry that dissociation is molecules coming apart, association is molecules coming together. One is the ligand, one is the receptor. The association rate is the rate constant times the concentration of the two molecules. And at equilibrium, we have the association and the dissociation equal, and the equilibrium constant is the products over reactants. So in the case of association, the products, ligand plus receptor, over reactants, which is this, is very high. So the association constant is very high for endocrine hormones. That's how they can find their specific targets, because they don't really bind very tightly to anything else. There are two different kinds of receptors for hormones. One is a cell surface receptor. Another is an intracellular receptor. So some hormones, like testosterone, are really hydrophobic enough to cross membranes. And they don't need to have their receptors on the outside because they can diffuse across the membrane. Their structures are shown here. So vitamin D, retinoic acid, cortisol, estrogen, thyroxine, which is made in the thyroid gland, and of course testosterone, which many of you are familiar with because of this phenomenon called testosterone poisoning. And this phenomenon occurs when guys get in fights in bars. So these bind to intracellular receptors, and then the intracellular receptors are actually induced to go and become transcription factors, amazingly. The hydrophobic ligand crosses the membrane, binds to its receptor. The activated receptor then becomes a transcription factor, binds to specific DNA sequences, and causes transcription. So all of these receptors have DNA binding domains. That's what these green boxes are. If the protein sequence is laid out linearly from N to C terminus, you can see a patch of protein sequence that binds DNA. And all of these act in the same way. They activate transcription. So those are intracellular receptors. Those are simple to understand compared to other types of receptors. There are other signaling molecules that can cross membranes. So one of them is nitric oxide. Nitric oxide is distinct from nitrous oxide, which dentists use, and it's called laughing gas. Nitric oxide is just NO. It's activated by an enzyme which takes arginine and puts it into and makes it into nitric oxide. This NO is not charged, therefore it diffuses across membranes easily. And it's an example of paracrine signaling. That causes guanyl cyclics to make cyclic GMP. So it converts GTP to GMP, cyclic GMP. And that causes a rapid relaxation of muscle cells. Interestingly, people were trying to develop a drug that reduces tension. It's an anti-hypertension drug. And they wanted to target this pathway. And specifically, they wanted to keep cyclic GMP alive for longer so that muscles and things would be more relaxed. So they made an inhibitor of phosphodiesterase, which converts cyclic GMP back to GMP to keep the cyclic GMP longer around. And they made this drug called Viagra. And when they tested this drug in clinical trials, it turned out to have another effect. And in fact, the men that were using it didn't want to give it back. It turned out to be one of the biggest selling drugs in history. Paracrine signaling is local, and this is the example of yeast having sex, as I was telling you before. That occurs during development and in other cases to form tissues. And neuronal or synaptic signaling occurs in the nervous system, where the action potential is propagated to cause the release of small molecule neurotransmitters to the target cells. In this case, the specific response depends on the neurotransmitter and the specific receptor for that neurotransmitter. And I'm going to go through an example of how one molecule can cause different responses because it has different receptors on different cells. At the nerve terminal, the electrical signaling, the action potential, the change in membrane potential, is converted to a chemical signal by calcium. Voltage-gated calcium channels open at the nerve terminal, letting calcium in. This causes the fusion of the synaptic vesicle, release of neurotransmitters. Neurotransmitters bind to the receptors, which can be ion channels. And I've talked about that in another lecture. So the reception on the cell is an electrical signal turned into a chemical signal, turned back into an electrical signal. And finally, to go through our examples of putting things in boxes, contact-dependent signaling is responsible for a number of cell fate choices when cells decide to become different things during development. And we're very interested in all these molecules because people want to make stem cells differentiate into various types of tissues. So this is contact-dependent signaling, where the membrane-bound signaling molecule interacts with the membrane-bound receptor. So, an example of how cells can decide to be different things or to respond differently is given by this response to acetylcholine. So acetylcholine is a neurotransmitter, which I talked about before in another lecture. Acetylcholine can cause muscle contraction in skeletal muscles. Okay, so I'm causing my skeletal muscle to contract. It can also cause relaxation or decreased heart rate in heart muscle cells. How can this be? It's because there's two different acetylcholine receptors, and I'm going to tell you about each one. Acetylcholine also causes secretion of salivary enzymes by the salivary glands. So if you think about, let's just all think about eating lemons right now. You put the lemon in your mouth, and you chew it up, and your salivary gland starts secreting stuff. Well, it's because acetylcholine is binding to the receptors of these cells, causing the release of all the enzymes necessary to take care of the lemon juice, that sour stuff. So, the nicotinic acetylcholine receptor is an ion channel, ligand-gated ion channel. I've talked about voltage-gated ion channels, which change conformation to let sodium or potassium through. This is a ligand-gated channel, which changes conformation to let sodium and calcium through. So acetylcholine actually binds to two different receptors. The one I talked about is the electrical signal converted into the chemical signal, the ion channel linked receptor. Okay? So it's a receptor that opens an ion channel. That's the first one. The second one, that one's the one that causes your muscles to contract, your skeletal muscles. The second one is a G protein coupled receptor. And that's the muscarinic acetylcholine receptor. It's a completely different receptor. Different gene, different mode of action. The ion channel receptor causes muscle contraction. In heart muscle, it's this muscarinic, binds to muscarinic G protein coupled receptor, causes heart muscle cells to relax and decrease their heart rate. So, G protein coupled receptors are the major class of receptors. There are hundreds of these in the body. So when we think about drugs that bind to them and all these pathways for signaling, we really have to understand G protein coupled receptors. They span the membrane 7 times. So one could call them serpentine because they look like serpents. Serpentine transmembrane, 7 transmembrane spanning receptors. GPCRs for short. They are in the plasma membrane. They're called G protein coupled receptors because they couple to G proteins. G proteins are the other type of molecular switch. They're GTP binding proteins. They're trimers, alpha, beta, gamma subunits. Alpha binds to GDP or GTP. Beta, gamma binds to alpha. And this is their structure here. Their mechanism of action is known. So this molecular switch, as I told you before, involves the active state being the GTP bound, not covalently linked, but just bound. GTP hydrolysis causes its inactivation. And then to reactivate it, it binds to GTP again. To cause inactivation, proteins help this process. Those are called GAPs, or GTPase activating proteins. To cause activations, these are called GEFs, or guanine nucleotide exchange factors. These cause GDP to be kicked out and GTP to bind. So, the theme of molecular switches. In contrast to kinases putting the phosphate on and phosphatases take it off, these are covalently bound, reversible. This one is non-covalently bound. GTP binds to activate the protein. GDP is made by hydrolysis to inactivate it. Okay? This is the way the G protein coupled receptor works. This receptor, the serpentine receptor, acts as a guanine nucleotide exchange factor for the trimeric G protein. The receptor activates the trimeric G protein, causing it to bind GTP. It goes off and it does its thing, signal out, and then the target protein activates hydrolysis, causing it to be inactivated, go back to its GDP bound state. So the target protein is a GAP, the receptor is a GEF, activates binding. So here's the cycle. We have the plasma membrane shown here with the gray lines, and the receptor in it binding to its ligand. So its single molecule binds to the receptor. That causes a binding site to open up on the receptor. So the receptor changes conformation, binds to the trimeric G protein on the cytoplasmic side of the receptor. The receptor acts as an exchange factor and kicks out its GDP, binds to GTP. There's a lot of GTP in cells because of mitochondria. So this is a favorable interaction. GTP binds, then the trimer dissociates. Activated alpha gets away from its beta gamma. Beta gamma is kind of like one subunit. There's two proteins. They're not covalently bound, but they act as one. So the protein splits apart. This guy can go on, activate something. This guy can go and activate something else. So you get, at this point, signal bifurcation. So let's take alpha. Alpha binds to its target protein, and it causes a downstream effect of some kind, activates the target. I'll talk about it specific examples later. Then the target acts as a GTPase activating protein, or a GAP. GTP is hydrolyzed, it dissociates, and now alpha is in its GDP bound state, goes back and binds to beta gamma, goes back to its resting state. So this is a whole cycle. Signal in, activation of this intracellular molecule, signal off. A G protein cycle. So we'll talk more about that in the next lecture of the signal transduction series. What I'd like you to think about when we're talking about signaling in general are some general themes. One is molecules turn on and turn off with a short half-life. So that ensures a rapid response. The cell resets the signal, so it's rapidly turned on and rapidly turned off. There are proteins that integrate a number of molecules called scaffold proteins, which keep everything in track. So there's a bunch of different pathways activated. It turns out there are proteins that bind to specific whole pathways, and that ensures that the signal doesn't just randomly and promiscuously do the wrong thing. Small molecules are made, and these small molecules act as intracellular second messengers to activate a whole bunch of downstream effects. That's called signal amplification. Signal bifurcation, alpha, beta, gamma, that's an example of two different signaling molecules starting from one event. So the signal bifurcates. So you get combinations of responses from one molecule. You also have the opposite. So the signals bifurcate, they also integrate into particular proteins, such as a transcription factor. You might need two or more signals to get a particular response. That's signal integration. And finally, sometimes you need multiple subunits to assemble all at once to get this kind of sigmoidal activation curve. That's called cooperative assembly of multiple subunits. So in this way, a small concentration in a signaling molecule can have a large effect on the protein. So when you're studying these signaling pathways, I know they're very complicated and it might seem overwhelming. Keep these principles in mind. And in particular, I'd like you to know the details of how G-protein coupled receptors work, the general principles of phosphorylation, dephosphorylation, and the G-protein cycle, molecular on-off switches, and know the different types of receptors used in signaling. So I'm going to stop this lecture here, and the next lecture will be the signaling